For a long time, we thought we had a decent map of the human genome. We focused on the big, obvious genes that code for the large proteins that build our bodies and run our cells. But we were missing almost an entire universe of information. Lurking in the vast stretches of DNA we once dismissed as junk is a hidden world, a dark matter of the genome, filled with tiny but powerful molecules called microproteins. And today, we're going to explore how the revolutionary gene editing tool CRISPR is finally allowing us to turn on the lights in this hidden biological world. What are these microproteins? Imagine them as miniature proteins, typically less than 100 or 150 amino acids long. For decades, the computer programs designed to find genes were built to look for long stretches of code, for the biological equivalent of novels. They were programmed to completely ignore the short poems and haikus. This meant a vast and crucial part of our proteome, the complete set of proteins in an organism, remained hidden in plain sight. These tiny proteins are translated from what scientists call small open reading frames, or SORFs, which are scattered all over our genome, sometimes hiding within other genes, sometimes in what we thought were non-coding regions. And their functions are incredibly diverse. They can act as fine-tuning knobs on larger protein machines, work as independent signaling molecules, or serve as critical components in fundamental processes like energy production. The first clues that this hidden world existed came from clever lab techniques. One of the most important was ribosome profiling. This method gives scientists a snapshot of all the RNA molecules being actively read by ribosomes, the protein-building factories of the cell. By capturing these tiny snippets of code being read, researchers could see that many of these supposedly insignificant small open reading frames were, in fact, being translated. But seeing a recipe being read isn't the same as tasting the dish. The field needed definitive proof that stable, functional microproteins were the end result. That proof came from a field called proteomics, specifically a technique called mass spectrometry. By analyzing the entire protein content of a cell, scientists could find the peptide fragments that perfectly match the sequences predicted from those small open reading frames. This was the smoking gun. The microproteins were real. But knowing they exist is one thing. Knowing what they actually do is the real prize. To figure that out, you can't just observe. You need to intervene. You need a tool to poke and prod the system. This is where the revolutionary gene editing tool, CRISPR, enters the story and changes everything. You've probably heard of CRISPR, often described as a pair of molecular scissors. It's a system borrowed from bacteria that can be programmed to find and cut DNA at a very specific location. Its precision and versatility have transformed biology. For the thousands of newly discovered microproteins, CRISPR provides a scalable way to systematically switch them off, one by one, and see what happens to the cell. One of the most powerful ways this is done is through something called a CRISPR screen. Imagine you're in a massive control room with 1 million unmarked light switches, and you want to know what each one does. You could start flipping them one at a time. A CRISPR screen does exactly this, but for genes. Scientists create a huge library of guide RNAs, the molecules that direct the CRISPR scissors. Each guide RNA is designed to target a specific microprotein gene. They then introduce this library into a population of millions of cells so that each cell gets its own unique knockout. One specific microprotein gene is disabled. By then observing how these different cell populations behave, scientists can infer the microprotein's function. For example, if cells with a certain microprotein knocked out suddenly stop growing, that tells you the microprotein is essential for cell proliferation. Or maybe the cells start storing more fat, pointing to a role in metabolism. Using modern DNA sequencing, researchers can quickly count which knockouts become more or less common under certain conditions, giving them a genome-wide map of function. It's a powerful and unbiased way to find the microproteins that truly matter. Let's look at some real-world examples of this in action, because this isn't just theoretical. Scientists at the Salk Institute were hunting for new ways to tackle obesity and related metabolic diseases. They created a custom CRISPR library to screen thousands of microproteins in adipocytes, or fat cells. By systematically knocking them out, they watched for changes in cell growth or lipid accumulation. They hit the jackpot, identifying dozens of previously unknown microproteins that act as regulators. One of these, which they named adipocyte SMORF1183, was shown to directly modulate how fat cells differentiate. This isn't just a cool discovery, it's a brand new potential target for developing future drugs to treat obesity. Cancer research is another area where this is exploding. Cancer is fundamentally a disease of uncontrolled cell growth, so finding the genetic switches that govern this process is a top priority. Several CRISPR screens have now identified microproteins that are essential for the survival and spread of cancer cells. For instance, one study found a microprotein called CASIMO1 that controls cell proliferation in certain cancers. Another identified a microprotein called HDSP that appears to promote the progression of gastric cancer. These discoveries are providing a whole new class of potential targets for cancer therapy. Many existing drugs target large, well-known proteins. These microproteins offer a chance to develop more specific therapies that might have fewer side effects. The fight against devastating neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's is also getting a boost from this new technology. In many of these conditions, the core problem is the toxic buildup of misfolded proteins in brain cells. Researchers are now using CRISPR screens, sometimes in neurons grown from patient stem cells, to hunt for microproteins that might regulate this process. Can we find a microprotein that acts like a tiny garbage disposal, helping the cell clear out these toxic clumps, or one that protects neurons from dying? The search is in its early days, but the potential to uncover entirely new disease mechanisms is immense. Of course, this research isn't easy. This is a new scientific frontier, and it comes with major technical challenges. For one, the small size of these microprotein genes makes them difficult to target precisely with CRISPR. There's a higher risk of the molecular scissors cutting in the wrong place, causing unintended off-target effects that could ruin an experiment. Scientists are developing higher fidelity versions of the CRISPR system to minimize this risk. Perhaps the biggest challenge is proving that the biological effect you observe comes from the microprotein itself, and not from the DNA or RNA that encodes it. A gene's RNA transcript can sometimes have a function all on its own. To solve this puzzle, researchers use clever experimental tricks. For example, they might use CRISPR to introduce a tiny mutation that changes just one letter in the genetic code. This change might be enough to create a nonsense protein, but it leaves the RNA molecule almost identical. If the biological effect disappears, you have strong evidence that the protein was the functional component. As with any powerful technology, especially one that touches our own genetic code, we have to consider the ethical implications. The discovery of this hidden layer of our biology further blurs the lines of what a gene even is. A stretch of DNA we thought was junk or part of one gene might actually encode a completely separate, functional microprotein. This complicates how we interpret genetic testing and diagnose diseases. And as we identify microproteins linked to health and disease, the temptation to use CRISPR to fix their genes will grow. This raises profound questions about the responsible use of this power, particularly when it comes to editing human embryos. We need a robust and ongoing public conversation about safety, ethics, and ensuring equitable access to any therapies that result from this research. So, where does this leave us? We have opened a door to a vast, hidden part of our own biology. The dark proteome is not dark because it's empty, but because we haven't looked there until now. What we're finding is a rich and complex landscape of tiny, functional molecules that play critical roles in health and disease. From discovering new drug targets for metabolic disorders and cancer to opening new fronts in the war against neurodegenerative diseases, CRISPR is the searchlight guiding us through this new world. The journey is just beginning, but the potential for discovery is limitless. We are embarking on a new chapter in biology, proving that sometimes the biggest discoveries are the ones you can't easily see. The future of biology, it seems, is small,